All right, video is recording, so I will go ahead and get started. I'm super excited that we have made it out of molecular biology and are now moving into more ecology stuff, evolution stuff, plants and animals. This is the stuff that I really think is, I don't want to say better, but at least more interesting for me. So hopefully you guys really enjoy it as well. So today we're going to be talking about evolution and how it works and some of the lines of evidence for evolution, as well as some of like the big picture concepts about how one species turns into two species. So we'll go ahead and talk about that. So in general, evolution is considered to be one of the unifying theories of biology, kind of how we ask questions about how the natural world works. It's a really important thing for us to understand if we want to ask questions about, the plants and animals that we're seeing in the natural world. So this idea of evolution, the fact that species are different now than they used to be, and some species don't exist anymore, that's been around for a while. That was, thrown around back in the 18th century by a few different naturalists. But really, the theory of evolution and the mechanisms of evolution is attributed to Charles Darwin. Although he and another scientist named Alfred Russel Wallace both came up with the idea back in the mid 1800s. Last lecture, we talked about how sort of two scientists or groups of scientists came to the same conclusion, but it was a little bit of a tense interaction. This was not a tense interaction. They were actually very nice to one another. They exchanged ideas in the series of letters. So this was actually, a nice, solid scientific friendship that developed. So this is pictures of the two of them down over here. They look very similar, but this one over here on the left is Charles Darwin, and then this one over here on the right is Alfred Russel Wallace. So both of them came up with this idea about the mechanisms of evolution, primarily through natural selection, which we'll talk about in a couple slides, in the mid 1800s. So when we talk about evolution, there's two types of evolution, and they're a little bit different. So we want to make sure we're understanding which process we're talking about here. So macroevolution is a lot of times what people think of when they think of evolution. This is big picture evolutionary changes that's giving rise to higher taxonomic groups. So something higher than a species, like new families of organisms, right? So this is sort of on a paleontological scale. So that's sort of what people think of when they think of evolution. But we are primarily going to be focusing on the mechanisms of microevolution. It's a little bit easier to track and to observe, so we're going to be talking about that one primarily. But we do want to keep in mind that macroevolution is a type of evolution. It's just happening on this really, really, really big time scale, but it's the same processes. It's just a lot of microevolution steps that have been smashed up together over a large time scale. So microevolution, when we're talking about microevolution, this is referred to as a change in allele frequencies in a population over time. So this is a very important definition for us to understand. So we're going to break it down so we understand what all of the different parts of this definition mean. So when we talk about allele frequencies, we talked about alleles in chapter 8. So this is the relative proportion of an allele. So how much of a certain type of allele is in a population. So how much of this gene variant is in a population, right? If we have these dominant alleles versus these recessive alleles, what's the relative proportion of the dominant compared to the recessive? So how are these alleles existing within a population? And when we talk about population, a population is a group of organisms of the same species that are close enough to breed. So they're living in the same place, they're of the same species, and it's that group of organisms. So that's what we're referring to when we're referring to a population. So this is a population of one type of species. And over time, so this is a small time frame, one generation to the next. So when we talked about macroevolution, that was a really, really large time frame. And when we're talking about microevolution, we can really observe it just one generation to the next. So a change in allele frequencies in a population over time is what we're talking about when we're talking about microevolution. Basically, what this is saying is that we look at one generation, we look at the next generation, and the way that we can see if evolution has happened just from one generation to the next is looking at those allele frequencies. So if we know genotypes in a population, we can know the allele frequencies in a population, and then we can compare one generation to the next. Has evolution occurred? Yes or no? We can tell that just by calculating these allele frequencies. There are a lot of different things that can cause these allele frequencies to change. We're going to talk about 5 of them. But before we talk about those 5 mechanisms of microevolution, we're going to just talk through an example of how allele frequencies might change. So remember that individuals carry 2 alleles of each gene, right? We have our genotypes: homozygous dominant, heterozygous, homozygous recessive. Those are the 3 types of genotypes we can have, and each of those have 2 alleles. So if we're homozygous dominant, we have 2 dominant alleles; homozygous recessive, 2 recessive alleles; heterozygous, 1 of each. Okay? So we've got 2 alleles coming from each individual. That's going to be important when we're trying to figure out how many of each allele is in a population. So if we have certain information about a population, we can calculate these allele frequencies. The example that we're going to use is the peppered moth. So this is a species of moth that is really helpful to kind of look at microevolution. There are 2 phenotypes that we see. We've got light pigmentation. You can see it's kind of speckled there. And that is a homozygous recessive genotype. So if we're talking about the letters here, we've got 2 little b's as our genotype. And then the dominant phenotype is the dark pigmentation. So that can be 2 different genotypes, right? That dominant phenotype can either be homozygous dominant or heterozygous. So we have 2 options of genotypes there. So this is the system that we're dealing with. So in our population, if we have a population of 10 moths and they have these genotypes that are listed right next to them, we want to try and figure out what the allele frequencies are. So how many of each allele is there? So we've got 2 different types of alleles. We've got big B's, we've got little b's. So we just need to figure out how much of each is present. So if we look at our big B's first, our dominant allele, if we count up all of the alleles there, we have 10 individuals, 2 alleles from each. That gives us 20 total alleles. If we just count up how many big B's we have 4, right? We have 2 big B's from this individual here, and then we have 1 big B from our heterozygous here, and another big B from our heterozygous there. So we've, out of all 20 of these alleles, 4 of them are the big B's. So 4 out of 20 is 0.2 or 20%. So that frequency is always going to be represented as a decimal or a percent, and it's going to range from 0 to 100 of our percents, or from 0 to 1 in our decimals. So this allele frequency here is going to be 20%. 20% of all of those alleles are big B's. If we do the same thing for the little b's, we can just subtract, right? If we know 4 of the 20 are capital B's, we know that the rest are going to have to be little, so that gives us 16. We can also just go through and count. We've got 2, 4, 6, 8, 10, 12, 14 little b's in all of our light colored moths, and then we have 15, 16 little b's from these heterozygous individuals. So 16 divided by 20, 0.8 or 80%. So these are our allele frequencies in the population. We can also do this with genotypes, right? Just like we did it with the alleles, we can do it with the genotypes. But instead of there being 20 alleles, we're going to be looking at 10 genotypes because each individual just has 1 genotype. So our homozygous dominant genotype, we can just count that right here. This 1 individual out of all 10 is going to be 0.1 or 10%. So 10% of our individuals are homozygous dominant. We look at heterozygous individuals. Our heterozygous individuals, we just have 2. So 2 out of 10 individuals, or 20%, are going to be heterozygous. And then our homozygous recessive here, we've got 7 individuals, so 7 out of 10 is 70%. Okay? So these are our genotype frequencies, different than our allele frequencies. So let's say that this peppered moth, and well, we don't have to say it, this is actually what happened. These peppered moths were living in an area in England before and during the Industrial Revolution. And before the Industrial Revolution, the trees that they lived on were had this white colored bark. So it was really easy for the predators like birds to pick off the dark colored pigmentation moths because it's much easier to see. During the Industrial Revolution, all of this coal is being burned, so all of these trees are getting lots of soot all over them. The trees are actually changing colors because they're just absorbing all of this color from the coal that's being burned. So now it's way different, right? These birds, instead of being able to easily see the dark colored moths, now they can easily see the light colored moths. So we're going to have this, before we had preferential predation on the dark colored moths, and now we have preferential predation on the light colored moths. So more light colored moths are being preyed upon than they were before. So that's going to change the number of light versus dark colored moths that we see. So now this is a little bit later. This is after these birds have had some time to prey on our light colored moths instead of our dark colored moths. And you can see that we have more dark colored individuals now. Instead of having just 3, now we have 5, and we only have 4 of these light colored individuals. So we still have our 10 moths with the genotypes that are listed there. So now we want to talk about our allele frequencies again. So we want to calculate our allele frequencies now to see if they've changed. So our big B, if we count up all of our big Bs, we've got 10 of them. So 10 out of 20 is 0.5 or 50%. And if we do that with our little b's, we also get 10 out of 20 or 50%. So this is different than the percents we saw before. So when we have this change in allele frequency over time in this population, we can say, yes, microevolution has occurred. So this is an example of how microevolution can happen and how we can track microevolution happening in a population. So let's talk about the 5 different mechanisms of evolution, how this can happen. A lot of times people only think of evolution by natural selection. That's kind of the one that Darwin and Wallace were championing, which is super, super important mechanism of evolution. But it's not the only way that allele frequencies can change in a population. There are a few ways that they can do that. So we're going to go through the 5 different mechanisms of microevolution here. So the first one is mutation. So this is a change in the DNA sequence of a gene. This is what we talked about in chapter 9, right there at the end, right? If we have a nucleotide that is changed or inserted or deleted, we can change the sequence of amino acids in our proteins. These proteins are coding for our phenotypes. So we can have new alleles created by mutation. Okay, so down there at the bottom in this chart, this is just showing that we have 3 alleles in a population. If we have a mutation that occurs, it can possibly lead to a brand new allele, right? We might not have seen this type of allele before. So this is going to be our source of new alleles. This is how we're going to get new alleles. All of our other ways that evolution works is working on existing alleles in a population. This is going to be where new alleles come from. So in order for these mutations to be important for evolutionary purposes, they have to be heritable. Right? If you have a mutation, but it doesn't get passed on to the next generation, that's not going to mean anything for evolution because it's not moving forward in the population. And overall, the effect of mutation on evolution is pretty small unless it's combined with some other mechanism, right? If this mutation leads to something that is makes you really successful in a given environment, this mutation, this new allele is going to increase in frequency. So it's going to need to combine with another mechanism in order to actually be have this large impact on evolution. Otherwise, it's going to be hard to get a lot of individuals with the same mutation increasing in frequency in the population. So this is what it would look like if we take away our pie charts and just look at beetles, for example. If we have we've got this population of green beetles, and we only have green beetles. If we have some sort of mutation that occurs, some change in DNA sequence, maybe we might get a change in the protein that codes for color. And now we have a brand new allele. We have an allele for brown beetle coloration instead of just green. So now we've increased the number of alleles. We have a brand new allele there. The second mechanism of evolution is what we call gene flow. So this is also basically it's just like migration. It's the movement of alleles into a population or out of a population. So there are 2 different ways that we can talk about this. Both of them are called immigration. They're just spelled differently. So if we're referring to immigration with an I, that means that individuals have moved into our population of interest, where we're tracking the allele frequencies. If we're talking about immigration with an E, that's referring to individuals leaving our population of interest, okay, and changing the allele frequencies there. So in our figure over here, we've got two populations of deer that are separated by a mountain range. If we have, let's say, all of these have, the same allele that's coding for coat color, and everybody in population 2 has the same allele that's coding for coat color. So if we have two individuals from population 1, from our western deer population, cross this mountain range and enter population 2, now we have a change in allele frequencies over here, right? Before we had 100% of alleles being coding for this coat color. Now we have a new allele that has entered the population through immigration and has changed the allele frequencies. So remember that when we're talking about microevolution, it's just a change in allele frequencies over time. So this has changed the allele frequencies here just by a movement of individuals into a population. The third type of mechanism we have is called genetic drift. So people always get genetic drift and gene flow confused. So make sure that you are able to differentiate these two. So gene flow is what we just talked about. That's migration. Genetic drift is something different. So this is a random event that causes the alleles in a population to decrease. Okay, so something is going to happen where we have our initial population. Our initial population is dramatically reduced. Our number of individuals is reduced. And during that reduction, we are losing some alleles. So if you are already a small population, this is going to impact you more because it's much more likely that you're going to lose these alleles. If you have a really, really, really big population and you dramatically reduce your number of individuals, you still might have a lot of individuals and have all of those alleles. So this is going to be impacting small populations more. There are two ways that genetic drift can happen. One of them is the founder effect. So that's what's shown here in the figure. So we have our big population of butterflies here on one island. The founder effect is where we have a few individuals leave that population and colonize a new area. And when that happens, it's just a few individuals, and it's possible that we might not get all of the allele variants that were present in the original population. So in our original population, you see we have 3 colors of butterflies here. We have, I can't tell if that's black or dark brown, but we have this darker colored butterfly, we have an orange butterfly, and a white butterfly. But in the founder effect, we have a few individuals that have left that population and colonized a new area, and we are missing that white phenotype, right? So we don't have all of the alleles that we had in the original population. So the founder effect, a few individuals leave the ancestral population, colonize a new area, one type of genetic drift. The other type of genetic drift is called the bottleneck effect. So this is where we have our large population and some sort of catastrophic random event happens that decreases the number in the population. So this could be maybe we have a field of flowers and a fire comes through and burns up most of the population and we're only left with a few individuals. That might be an example of a bottleneck. A lot of times what we see with mammal species, if mammal species have gone through a bottleneck, it's because of maybe like predation by humans. So like cheetahs, they don't nearly have the genetic diversity that they used to because they were hunted a lot. So that's an example of a bottleneck, a large population going down to a small population. So there are a number of ways that can happen, but it has nothing to do with the genotypes of an individual, right? Certain individuals aren't able to survive more than another. This is a completely random event that is just going to decrease the number of individuals in a population. So here we've got some individuals up in our original population that have lots of different, these 3 different types of genotypes that we've been seeing, homozygous dominant and recessive and heterozygous. Something has happened here where we have decreased the number of individuals to just a few, and we have lost that dominant allele, right? So now we have changed the frequency of alleles in this population. We no longer have that big B letter, that big B allele. So we've changed the allele frequency in a population and microevolution has occurred, and it's occurred through a bottleneck of genetic drift. The fourth type of mechanism, this is the one that Darwin and Wallace really, really talked about, and this is probably the one that most people think of. It's a process where a heritable trait makes it more likely that an individual can survive and reproduce in the current environment. Okay, there's something that makes you better fit for an environment and makes you survive and reproduce more than other types of individuals. So down here, we've got an example here, population of beetles that are green and brown. Something about the environment here in this population makes it so that predators can more easily prey on the green beetles. So what's going to happen to the green beetles? They're going to decrease in frequency, right? You're going to have fewer individuals. These brown colored beetles are able to survive and reproduce more than these green beetles. So over time, we're going to see a decrease in the number of green individuals and an increase in the number of these brown or tan individuals. So change in allele frequencies over time, change in allele frequencies over time due to natural selection. So this example was predation, but it's not just predation. Natural selection can happen for a few different, in a few different ways. Disease is another one. If there's something about your genetic makeup that makes you resistant to some sort of disease, you are more likely to survive and reproduce in an environment where disease is present. Environmental conditions might be important, right? So if you are living somewhere that's really, really cold and you have thicker fur and that is heritable, you can pass that on to the next generation. That trait is likely to increase, right? If staying warm allows you to survive and reproduce in a current cold environment, that's going to be a way that natural selection can work. So it's not just predation. There's lots of ways that natural selection can happen. In order for natural selection to happen, there are a few things that this trait has to have. First of all, there has to be variation in traits, right? If everybody's the same, then no individual based on that trait is going to be able to survive and reproduce more than anybody else. So we have to see some variation. These different traits also have to have differential reproductive success, right? If you are different than somebody in your population, but that doesn't matter at all for surviving and reproducing, you're not necessarily more fit because you have a certain trait, right? And when we're talking about fitness here, we're talking about an organism's ability to survive and reproduce. Okay, so we have to have this different levels of fitness corresponding to this trait variation. And then that trait also has to be heritable. Okay, so even if you have a trait that allows you to survive and reproduce more than other individuals in the population, if you can't pass that on to the next generation, there's nothing for evolution to work on there. So your traits have to be variable, they have to have different levels of fitness, and they also have to be heritable. So we have to have those three things in order for natural selection to work. And then the fifth mechanism of evolution is sexual selection. So this is a type of natural selection, but it's just referring to mating, and it's referring to mating that is not random. So over here in our beetle figure on the left side, when we have random mating, this individual is just as likely to mate with this beetle or this beetle or this beetle. Doesn't matter. Maybe it's whichever one they come across first. Maybe it has to do with something else, but it has nothing to do with this variation in trait. Non-random mating or sexual selection means that this individual is preferentially mating with this individual compared to the other two. Okay, so non-random mating is sexual selection. Okay, and a lot of times with sexual selection, we see traits that are increasing reproductive success, but not necessarily increasing survival. It's one of these really strange things about sexual selection, right? We think of a peacock. That giant tail that a peacock has does not help it survive, right? It actually makes it harder for it to survive. Yet this giant tail is increasing their reproductive success. So you kind of get a little bit of a clash. It's not necessarily helping you survive, but it is helping you reproduce. So, what are the pros and cons of that interaction there? So we get some really interesting things that are some traits that are caused by sexual selection. A lot of times, any sort of giant ornamental trait you see on an organism, a lot of times that's going to be due to sexual selection. And a lot of times it's due to some sort of female preference. You see these large ornaments on male individuals. Females are preferentially choosing males with that large ornament to reproduce with. And there's lots of research out there. Why would a female choose this ornamented male over this other male? And maybe it is the fact that that ornament makes it harder to survive, right? If this giant peacock tail makes it hard for a peacock to survive, but that peacock's still alive, maybe that female is like, oh, well, he's probably got some pretty strong genes if he can survive with that giant tail, right? There's lots of theories out there about how this works. Super interesting stuff there. Okay, so if we have selection happening, there are three types of outcomes that we can see from this selection that's happening. So up here at the top, we have just our normal distribution of a trait, right? Remember, we have variation in a trait in a population. So in this example, we've got different colors of mice. So we've just got a normal distribution, this bell curve trend, normal distribution there of our traits. So there are three different ways that selection can introduce this distribution of traits. Our first option is the first one over here called stabilizing selection. So stabilizing selection is going to be where something about this middle type of trait is going to be better and have a higher fitness. So this one is going to increase, and the extremes of our bell curve here are going to go down. So we are going to sort of cull this extreme variation on the sides and narrow our distribution. Stabilizing selection is that one. Directional selection is where one extreme is favored over the other. This is kind of what we saw with the peppered moth example, right? Our light colored individual was favored or was able to survive and reproduce more before the Industrial Revolution. The darker colored individual was able to survive and reproduce more after the Industrial Revolution. So we got this shift, this directional shift in the curve of natural traits that we see. So it favors one extreme and shifts the distribution either to the right, or to the left. Okay, so we still have a wide variation in traits, but it's just shifting it one way or another. That's directional selection. And then disruptive selection, kind of the opposite of the stabilizing selection, where rather than the middle being favored, the extremes are going to be favored. So we're going to see a decrease in that middle trait there, and an increase in the extreme traits, the lightest color and the darkest color in this example of mice. So this is going to create what's called a bimodal distribution, just meaning that it's got two peaks. Okay, so this bimodal distribution there is going to be the result of disruptive selection. So stabilizing, directional, and disruptive selection are the three outcomes that we can have, and it kind of shifts the curve accordingly. So now we're going to move on from the mechanisms of evolution and talk about the different lines of evidence for evolution, and there's a lot of them. So the first one is going to be fossil evidence. So this is basically just saying that when we find fossils, they look different than what we see today, right? Organisms have changed. They're not the same today as those that were found in the past. So if we're lucky enough, sometimes we can get fossil records that show the progression of evolution. A lot of times this isn't a perfect progression because it's really hard to make a fossil. It's really, really rare. So we don't oftentimes have every piece, but a lot of times we can kind of still see the variation over time that we've seen and the changes that have occurred. One example, a good example that we have is actually horse evolution in North America. So we had a change in the environment, went from a more forested area to a drier plains type environment. So with that change in environment, we had a change in anatomy. So there was a change in teeth shape and a change in the foot and the leg anatomy. And so we can see we've got fossils back from about 55 to 45 million years ago all the way to recent. And we can see that these individuals look different. They look different in their teeth. They look different in their legs and their feet. And this is most likely a result of evolution by natural selection. The environment is changing. Different traits are going to be favored in these newer environments. So that trait is going to proliferate in the population and move on in the next generation and be passed on to next generations. So this is a sort of backing up to a much more larger scale of evolution, but it's still the same thing. We're seeing this change in allele frequencies, but we're big time scale here looking at these changes in traits. We can also sometimes find transitional fossils that can show these evolutionary links. So a really great example of this is the fossil Archaeopteryx. This was found in the mid 1800s. This is thought to be sort of the transitional fossil between dinosaurs and what we think of as birds today. Really, the birds that we see today are just modern versions of dinosaurs, but they have some different traits than dinosaurs did. And this Archaeopteryx, which multiple of them have been found, they have traits of birds and they still have some traits of dinosaurs. They've got teeth, they've got a tail that has vertebrae, but they've got wings and feathers like birds do. So there's this really great video about feather evolution. We're not going to watch it now, but I just wanted to link to it here, and I'm going to post it in the D2L folder for this chapter so you guys can watch it. It's narrated by Carl Zimmer, who's a really excellent evolutionary biologist. So I'll post that video for you. The second line of evidence we're going to talk about is biogeography. So this is just referring to how organisms are distributed on the planet. Okay, so the geographic distribution of organisms. So one aspect of biogeography that shows evidence for this is that broad groups evolved before the continents split up. So we had, Pangea, that was this amalgamation of continents before they split apart to what we know our continents to be today. So this example down here is this type of flower. So this type of flower is seen in all of these environments down here, all of these red colored areas here. This flower is seen in all of those areas. So that is evidence for the fact that this flower evolved when the continents still looked like this and we're all kind of bunched up together, because it's really easy for an organism to spread and increase its range when everything's so close. This is almost impossible for an organism to spread its range across, right? If you have an organism over here, it's going to be virtually impossible for that organism to naturally get to this other continent. So this flower evolved when all of these continents were still all pushed together. But we can also look at biogeography and say that the groups that have evolved since this split, so after the split, are going to be unique to a region. So one of the best examples of this is Australia. Australia has been isolated from other continents for a really long time. And so because of that, it has a lot of organisms that are found in Australia and not found anywhere else. So when we talk about a species that's found in one area and not found anywhere else, it's referred to as endemic. So Australia has many endemic species. And we can think of the marsupials that Australia has. Other places do have marsupials, but they don't have the marsupials that Australia has, right? They don't have koalas or kangaroos or wombats. My battery's running low. Give me 1 second. I gotta plug in, and then we'll talk about Australia a little bit more. Okay, so Australia has been alone for a long time, so it's been able to have all of these species evolve, and since it's so isolated, they're not able to move to other places, right? So this has a lot of endemic species, and there are other examples of this as well. Madagascar is a good example. That's an island off the coast of Africa, has a lot of endemic species as well, and we see this happening in islands primarily, but can happen in other geographically isolated areas also. So the third line of evidence is molecular evidence. So when we're talking about the molecular evidence, we can see evidence of a common ancestor just by the fact that everything has DNA, right? So that's evidence for a common ancestor of all life right there. Everything has DNA. Everything goes through that central dogma that we talked about in chapter 9, DNA, RNA, proteins. Everything does that. Those are all similar processes. So that's evidence right there on the molecular level. But we can also look at how different groups are related if we compare how similar their DNA sequences are. So this figure over here, we're comparing the percent of amino acids in human hemoglobin and comparing that to hemoglobin in other types of organisms. So when we compare a human to a rhesus monkey, the rhesus monkey has 95% of the same amino acids in hemoglobin that we do, right? That's very, very similar. So we can say that these two types of organisms are more closely related. And then kind of as we go down this list here, we're getting further and further away on the evolutionary tree. So we're getting fewer and fewer amino acids that are the same. So we can see how related things are based on, the similarities of their DNA sequences in those organisms. The fourth line of evidence is anatomical evidence. So there are a few different types of anatomical evidence that we can look at. One of those is called vestigial structures. So this is a structure that's basically like a leftover structure. It's not really used anymore, but the only reason we have it is because our ancestors needed it. Okay, so this is a structure that is appearing non-functional, left over from a past ancestor. So a great example of this is the fact that bones or whales have pelvic bones, right? They don't have back legs, so why would they have pelvic bones? These are remnants of a four-legged organism, right? So whales and dolphins evolved from an organism that had these back legs, but over time, it was much more beneficial for them to be more streamlined and dedicate the rest of their body, that caudal end of their body, to swimming. So they didn't need those back legs anymore. So eventually they were lost, but these pelvic bones still exist. Okay, so we have these leftover pelvic bones in whales that are left over from a past ancestor. Humans have a lot of examples of vestigial structures. One of them is our appendix, right? We don't actually use our appendix anymore. It's left over from an ancestor that needed to digest plant matter a little bit more than we do now. So we don't use it, but it's still there. We also have goosebumps. Goosebumps are a vestigial structure as well. So that's left over. So a lot of times when you see this in birds and mammals, if they're in a cold environment, they need to puff up to help keep them warm. So if you get puffier, you can trap air close to your body, and then your body heat can warm up that air and keep you warmer. So goosebumps are a vestigial structure of that, right? So it's these little, right around the hair follicle. When it's cold, it causes the hair to stand up. Okay, so goosebumps, vestigial structure in humans, and then wisdom teeth are also a vestigial structure. We had a common ancestor that needed that extra tooth back there to grind and tear things up. We don't need it anymore, but we still have them. It's actually really interesting because we are losing our wisdom teeth, actually. There's not necessarily an evolutionary pressure for us to lose our wisdom teeth, but we do see that. There are a lot of people who have all four wisdom teeth and need to get them all removed. There are some people who don't have any wisdom teeth. I think it's like 35% of people now don't have all of their wisdom teeth, right? I only had like 1.5 wisdom teeth. So we're kind of in this weird transitional period where we're actually losing this vestigial structure. Another line of anatomical evidence is homologous structures. So these are structures that share a common ancestor and have the same overall construction. Okay, so the classic example of homologous structures is the mammalian forearm. If we think about mammals, we've got a wide variety of mammals that have a wide variety of forearms. Right? We've got humans with our hands. We've got cats with their paws, whales and bats. These are widely varying structures, but all share a common ancestor, so all have the same overall construction. So they've colored the bones here in this figure to show you this same overall construction. So the humerus is there in purple, radius and ulna are there in orange and tan. Then we've got our wrist bones, and then our hand and our finger bones as well. Okay, so we can see that these have the same overall construction, the same types of bones, they're just laid out a little bit differently. So this shows that we had one common ancestor and it diverged into multiple structures that have the same overall construction, homologous structures. And then the last line of evidence is convergence. So convergent evolution is where we have organisms that are under similar pressures environmental pressures, predator pressures, whatever it may be. And because of those pressures, they evolve structures that are similar. So if we think about Arctic foxes and ptarmigans, that's this bird over here, both of these are white. This has nothing to do with the fact that they share a common ancestor, but they both live in the Arctic and it's white. So it's beneficial for them to also be white, right? They can hide in this white, snowy environment if they are also colored white. So they don't necessarily share a common ancestor that was white and now they're both white. It was this evolutionary pressure that caused them to evolve similar structures. Okay. This is a couple more examples, but I wanted to say that convergent evolution is creating what are called analogous structures. Okay. We talked about homologous structures. That's different. Convergent evolution is creating analogous structures. So these are similar structures from distantly related groups. So our homologous structures were different. Our analogous structures have, it's the same structure that has evolved multiple times in different groups. So we can think of wings. We can think of all of the flying animals you can think of. Insects, birds, bats, okay? All of these types of organisms have wings, but all of these wings evolved separately. Insects evolved wings, birds evolved wings, bats evolved wings. It was multiple evolutionary events that led to this creation of wings in these different lines of species. We can also think of animals that live in water, aquatic animals. They all have this really streamlined body shape. Sharks, we've got ichthyosaurs here, penguins, dolphins. So, we see animals that have evolved to live in aquatic environments have this streamlined shape. And that doesn't say that there was one aquatic ancestor that gave rise to everything that lives in water. No, things evolved to live in water multiple times, right? Birds came out of the water and then penguins went back in. Mammals came out of the water, dolphins and whales and seals went back in, right? So wings, body shapes of aquatic animals, these are going to be analogous structures, a result of convergent evolution. So just to clarify the difference between homologous structures and analogous structures, these get confused a lot. So I want to make sure that we understand the difference here. So when we have homologous structures, we have one common ancestor that has evolved multiple different types of structures. And when we have analogous structures, we have multiple evolutionary events that have converged on one structure. So streamlined appendages in our aquatic animals here, right? This fin and this wing and this flipper all look the same. And then again, we have our mammalian forearms here as our classic example of homologous structures. So it's very important that we understand the difference between homologous and analogous. Okay, so we're going to move on from our lines of evolution or lines of evidence for evolution, and we're going to talk about speciation. So far when we've been talking about evolution, we've talked about microevolution, right? That change in allele frequencies over time. But we're going to sort of back up from the population level and get a little bit bigger and talk about the species level. So this is a bigger scale that evolution can happen on. So speciation is where we have the formation of two species from one species. We've got one species that splits into two. So this figure down here, this is a page, a very famous page out of Charles Darwin's notebook that kind of illustrates how he thought speciation happened and how the tree of life looked, right? We have one species here that branches off into multiple species, and maybe some of these die out along the way, and we might be left with A, B, C, and D, like the four species that we're looking at. So they are all a result of these multiple instances of speciation. So in order to talk about speciation, we have to talk about a species. Species is a little bit trickier to define than we would like, but generally we operate off what's called the biological species concept, which is where we define a species as being a group of actually or potentially interbreeding individuals. So our population was a group of actually breeding individuals, but if we have multiple populations, they're not necessarily not interbreeding because they're a different species. They might just be far away and they can't, but if they were close to each other, they could interbreed. So that's where that potentially comes in. So actually or potentially interbreeding individuals. So this overall is pretty decent for sexually reproducing organisms, which is the majority of what we're working with here.
But there are some weird parts of it, right? What about hybrids? There are instances of blue whales and fin whales mating and producing a hybrid individual, right? If you've been watching Tiger King on Netflix, you know that tigers and lions can interbreed and create a hybrid. So maybe what's happening there is that speciation just isn't perfectly complete yet, right? Maybe we're nearing the end of it, but it's not actually complete. So they can still technically interbreed. Maybe that's what's going on there. So that's kind of one of the ways that we explain away hybrids when we're talking about the biological species concept, because speciation doesn't just happen in one instance. It's a long process. So if we've caught it in the middle of that process, there's still a lot of potential for interbreeding there. There are two types of speciation. The first type of speciation is allopatric speciation. This one usually makes the most sense when we talk about it, right? So we have a population, it gets separated by some sort of geographic barrier, and then they can't cross that barrier. Eventually, over time, they evolve to a point where they are no longer able to interbreed and reproduce. Okay, so we have our original population that's separated by a geographic barrier. That geographic barrier causes reproductive isolation, which eventually results in speciation. And then this green circle and this blue circle, those populations are no longer able to interbreed. So that's allopatric speciation. There are two ways that allopatric speciation can happen. So it's possible that we can have dispersal, which is where a few members of that species move to a new area. So let's say we've got species A over here with some variation in traits, and they are inhabiting an area that's right next to a river. Something happens. Maybe this is a species of flower, and there's a really big windstorm, and something gets blown across to the other side of the river. So this windstorm is like totally rare. We don't see wind speeds like this in this area often, so it was extremely unlikely for this species to get over here, but they made it. Okay, they can't cross back and forth easily. This is a reproductive barrier, geographic barrier there. So over time, we've got this new species, or species A prime, developing across the river, and eventually that could develop into a brand new species. So we still have our original species over here, but now we have another one on the other side of that river. Vicariance is the other mechanism for allopatric speciation. This is where some sort of natural event causes a physical separation. So rather than our species being next to a river, let's say we're occupying an area and something happens to where we have the formation of a river coming through our habitat area. So that breaks us apart into two different groups, and eventually we can have reproductive isolation. Even if that river disappears, we've got two brand new species. So geographic barrier is causing this reproductive isolation in allopatric speciation. An example of this is the spotted owls in North America. So we've got the northern spotted owl and we've got the Mexican spotted owl. So you can see that they occupy different areas, different geographic areas. And it's hypothesized that these were originally one species, but they were separated, possibly by the presence of glaciers during the Ice Age. So the glaciers came down this way and then retreated. But by the time they retreated, there had been enough evolution happening in these two different groups that now they are different species. Okay, geographic isolation resulting in reproductive isolation. We can also see allopatric speciation happening in something that's called adaptive radiation. So this is basically where we are getting many, many adaptations that are evolving rather than one species that is just splitting into two. This is one species that's splitting into several new ones, not just two species, but many species. The classic example of adaptive radiation is the Galapagos finches. So we start with an ancestral ground-eating finch, so a bird that ate seeds, and then there was adaptive radiation that happened, and now we have all of these types of finches that have variations on their beak shapes that allows them to eat different things. Some eat mainly insects, some eat mainly seeds, some eat buds and fruits, some eat cactus seeds and cactus parts, right? So we've got this adaptive radiation that happens there. The second type of speciation is sympatric speciation. This one is a little bit harder to think about because there's no geographic barrier that's happening. So there's still two separate species that are occurring, but it's happening, reproductive isolation is happening within a population. So no geographic barrier in sympatric speciation. One good example of this, we've got some cichlid fishes. This should say fish. I don't know why that says Sai, but two species of cichlid fish live in the same lake in Nicaragua. So there's no geographic isolation here. They're all in the same lake, but now they have different morphologies that allow them to eat different types of food. So something happened where one group of this population started eating one type of food, the other part of the population was eating another type of food, and so now they have different morphologies that allow them to specialize on these types of food. So we have two different species that are being created, even though they were never actually geographically separate from one another. So once these species are separate, how do they stay separate? So we have certain reproductive isolating mechanisms that prevent successful mating between the species. Okay? And by successful mating, we're referring to fertile offspring. So that's what we're looking for when we're talking about a successful mating. So we have these mechanisms in place to help species stay separate. So these mechanisms can be pre-zygotic or they can be post-zygotic. So if we remember talking about a zygote, right, that unicellular diploid structure, that's the result of fertilization. So some of these reproductive isolating mechanisms happen before that zygote is formed. Some of these reproductive isolating mechanisms happen after the zygote has already been formed. So pre-zygotic and post-zygotic. So one type of pre-zygotic isolating mechanism is temporal isolation. So this is when we have two species, but their mating times are at different times of year. Okay, so they're not going to mate with each other because they don't overlap in time. So an example of that is frogs. Maybe we have these two species of frogs that live in the same pond, but they breed during different seasons. Maybe one breeds in the summer, one breeds in the spring. So they're not going to breed with each other just because it's happening at different times of the year. Another type of pre-zygotic isolating mechanism is ecological. So this could also be referred to as habitat isolation. So two species could potentially interbreed, but they occupy different habitats. So this is why we don't see ligers in the wild, right? Lions and tigers can interbreed, but they usually occupy habitats that are different. So they're not encountering each other in nature, so they're not going to interbreed. We can also have behavioral isolation. So we have two species that live in the same place and maybe even mate at the same time of year, but they have different behaviors associated with courtship, and they're only going to respond to the behavior of their own species. So an example of this is birds. Certain groups of birds only respond to mating calls of their own species. So they might be hearing other birds making mating calls, but they're not going to respond to those because it's not the right call. And so that's behavioral isolation. And there's lots of really cool examples of behavioral isolation. You see lots of really elaborate mating behaviors. Like in the Galapagos, there's a species of bird called the blue-footed booby that does this really cool mating dance. And that's sort of a behavioral isolating mechanism because not every type of bird is going to respond to that dance. Mechanical isolation is another type of pre-zygotic isolating mechanism. So this is a physical difference that is going to prevent copulation or pollination. Okay, so this is like a lock and a key situation, right? If you don't have the right key to fit in that lock, then nothing's going to happen. Okay, so the example of this given here, certain breeds of dogs are morphologically not capable of mating due to size, right? We've got Great Danes, we've got Chihuahuas, not morphologically possible. So mechanically, they are isolated from one another. So that's another way that species can be separate and stay separate in a pre-zygotic mechanism. We also have a few post-zygotic isolating mechanisms. So we're going to go through those here. One of those is hybrid inviability. So remember, post-zygotic means that the zygote has already been formed. They have already mated, and our zygote is formed. But now there are mechanisms in place to prevent this offspring from surviving and reproducing and being fertile. So hybrid inviability is when hybrids are produced, but they don't actually survive. They don't develop to reproductive maturity. So an example of this, we've got certain types of frogs. They are able to interbreed and produce tadpoles, but those tadpoles never actually become frogs. So those hybrids are inviable. They don't develop to reproductive maturity. Another example is hybrid infertility. So our hybrids are produced, they are developing to reproductive maturity, but they don't produce gametes that are functional. They are sterile. So the classic example of this is a mule, right? A mule is a result of a mating between a horse and a donkey. But this hybrid mule, is infertile. They are sterile. You can't mate a mule with another mule and get more mules. It's not how it works. They're completely sterile. So this is another post-zygotic isolating mechanism, this hybrid infertility. And then the last one is called hybrid breakdown. So this is where our hybrids are fertile, but then, and they might even be able to produce gametes that are functional, right? Like they're fertile, they're producing these functional gametes, but when these gametes combine, that generation fails to develop. So the offspring of hybrid, so this is talking about copepods. These are plankton that live in the ocean. So copepods are not able to survive and reproduce. So this individual itself is a hybrid, but it can't create offspring that are able to survive and reproduce. So that's called hybrid breakdown. Okay, so all 7 of these isolating mechanisms you're going to need to be familiar with, understand how each one works, pre-zygotic and post-zygotic. So make sure that you are familiar with all of those isolating mechanisms. So that's all I have for you guys today. So I will get this uploaded so you can rewatch it if you need to have some clarification on different parts of the lecture. If you haven't marked yourself as being here in the chat, please do so. I think I almost got everybody. Maybe there's 1 or 2 people that haven't popped in there. So I'll keep the chat open for a couple more minutes. Make sure you mark yourself as here in the chat. And then also today, make sure that you get homework number 9 submitted. I posted a video exactly detailing how to go over every single problem in homework number 9 to make sure that you guys understand that. So please take a look at the video if you are confused on how to complete those problems. And make sure you get it turned in on D2L. All right, well, I'm going to go ahead and stop the recording, but I will be here for a couple more minutes if you guys have any questions. Thanks for coming.